не понимаете, сразу можете поднимать руку или писать в чат, хорошо?
Perfect. Okay, great. I think we can start. Welcome.
Does everybody see presentation? Yes.
Yes.
Cool.
So, dear CCFCA members, welcome to the webinar. This time dedicated to the coronavirus vaccines. And we invited this time we invited Asel Musabekova. It is already the second time when she's presenting the webinar. The first one was, I guess, in summer, and Asel gave us the analysis of coronavirus situation in France and in Kazakhstan. Asel is a PhD molecular biologist and virologist. She defended her PhD at the University of Strasbourg, and now she works as a volunteer at MedSupport KZ. It is the medical channel in Instagram, and she's a consultant for UNICEF Kazakhstan. She has a blog about science on Instagram, Asel Virology, to popularize science. So, Asel, the floor is yours.
Okay, cool. Thank you very much. I'm very glad to be here again. I hope you're all well, and let's start. So today we're going to speak about the vaccines against COVID-19. So vaccines actually against the virus, so SARS-CoV-2. And yeah, I have done some work on actually making this complicated science of vaccinology a bit more clear for general public in Kazakhstan. And I think that this year, all of us have almost received a degree in vaccinology, so which is really good news for me. Let's discuss now. We have a lot of questions, right? Because today was a record of new cases by day in Kazakhstan. It was more than 1,400 people having new cases, and this is a record since 6 months. So it may probably because of the testing, but also, of course, we see the second or the third wave, however you want to call it, is rising. Yeah, and we're going to expect more cases this coming months. And there are many questions about vaccines. So which vaccine to choose? What about the mutations of the virus? What are the RNA vaccines? What about the immunity? What kind of immunity do we want to expect from the vaccine? So I will try to answer some of these questions. Excuse me, I'm going to cough a little bit today because I'm having this residual cough. I'm very sorry for that. Now, when we speak about vaccine development, so first of all, we start with preclinical studies, of course. And vaccine development history was built on blood of people, literally. And the regulations that we have now for the safety of vaccines are of the highest standard because of this of really vigorous approach to testing vaccine efficiency and first of all vaccine safety. And this I want to really emphasize, that we cannot compromise vaccine safety guidelines, even in the time of the pandemic. This is my subjective, my own opinion, and I will explain you why. So vaccine development starts, of course, from preclinical studies. What is preclinical studies? It's when we speak about cell cultures, or human or animal cell cultures here, and then we have laboratory animals. Then we have, of course, we have to test them in primates. And then we move on to clinical trials where we have phase 1, which is mainly safety. So first of all, before even testing the efficiency, we have few people up to it depends, right? For the COVID trials, it was actually less than usual, but usually it's up to 100 people or a bit more. Then we have phase 2 trial where you have several hundreds of people, you expand. So this is called expanded safety trial because you expand this to the population of some vulnerable people, for example, population of certain age, some population groups with risk factors, etc. Right? So you not only have their healthy young adults, but also some other populations, and this expands to several hundreds of people. And then you have phase 3 trial where you know that we are having many thousands of people. As for COVID-19 trials, you will see later for the numbers, but it was up to 70,000 people, which is actually quite a good number for phase 3. And then the vaccines are being approved, and this is very particular situation in the COVID-19 trials, and we will speak about this later as well. There is also so-called phase 4 trial, which is not a trial, but the monitoring of vaccine safety and efficiency after the registration. So let's speak more about this. We need to speak about some of the exceptions which have happened during the pandemics. So first of all, there is such thing that early or limited approval. So basically, some countries, especially we're speaking about China first, who started this with limited approval of their vaccine already in June, and then Russia, of course, with Sputnik 5, which was approved also quite early before the completion of the 3 trials. And this is the situation where some people argue that this is possible because it's the time of the pandemics, and given the demand of the vaccine, this is acceptable. Then we have such thing, we also witnessed it during this period of COVID trials, that we have combined phases. And for most of the candidates, we have seen this, and this was combined phase 1 and 2, where actually, so the goals, the milestones for these 2 trials were combined into one. I don't particularly like this because I think this is 2 different things, right? For example, for the Sputnik trial, the phase 3 started even before the completion of the combined first and second phase. So we're going to discuss it later after the presentation, and I would like to hear as well your opinion. And then, of course, post-registration monitoring. And this is here we can think about an example of a Pfizer-BioNTech vaccine, where Pfizer have been approved for by the UK agency, and then on the first day of the vaccination, you know, you've probably heard about this, that there were allergy reactions which were not monitored during the trial because people with these allergies were not accepted to the trial. That's why that was actually the reason for the FDA to agree on the registration of Pfizer, but only on the condition that there's going to be post-registration monitoring during 2 years, which is, I think, a very good decision. Okay, now this was an overview of how we test the vaccine, and I want to emphasize that this process have been done. So it was put there for a reason, right? There is really a good reason to have this complicated, expensive, requiring 1,000 people trials for vaccines. Now let's speak about coronavirus itself. Of course, you all know it, but just to remind you that virus is basically genetic information packed into a protein shell. In this case, we have viral RNA, which is packed into shell of glycoproteins, and then we have the spike proteins. And these spike proteins are encoded by this part of RNA. And these spike proteins are actually the main thing that is being targeted by the vaccine manufacturers. Why? Because basically, we can say that the spike protein is the key. If you imagine that a human being here is a virus, and the spike protein is a key, so this door is actually a receptor on a human cell, which is called ACE2. And this receptor binds this spike proteins, they change conformation and they make virus enter into the cell. So this is the key for the virus to actually fuse with the cell. Now I'm going to show you this very funny video. I find it very cool. It's actually very realistic in terms of biology. So here you have the spike protein and it has several domains. It basically, you see this handshake. Basically, this is the key and the door. This is the ACE protein on the surface of the cell, and then this handshake, it enables the virus and the cell to fuse. It's cool, right?
Yes. Well, we have three rules, and our philosophy was if we can do it here, we can do it anywhere. Basically, right?
I'm sorry. Somebody is talking, right? Do you want? I didn't. Was that a question? Hello. Okay, somebody didn't mute the microphone, probably. Okay, let's continue. Did you all see the video?
Yes.
Yeah, you agree that it's cool, right? Okay. Let's see. So now we have different approaches to vaccines. We have the traditional approach where we have the whole virus, and this virus can be live or inactivated. The thing is that the live virus vaccines are not really preferred because there are a lot of risks with having an actual virus which will be able to cause a disease. That's why in the whole virus vaccines, we actually prefer the inactivated virus approach. So what is it, inactivated virus? So we have the virus and then we treat it with some sort of chemicals. In this case, in some of the candidates, we saw that it was treated with beta-propiolactone. So what are these chemicals? These chemicals are actually making this virus not able to infect and to cause a disease. How? This chemical binds the RNA inside the virus so that it's unable to reproduce and to actually cause the inflammation. Now, let's see what happens then. So we have this inactivated virus which enters the cell, which fuses with the cell, just like the virus that we saw now, just like the live virus. And then the cell which is infected, it will produce these spike proteins or some other antigens, and it will expose it on the surface of the cell. And like that, our immune system will produce antibodies against these parts of the virus. So this is very logical, very old approach. So basically, the main advantage of this is that this is a known technology. And also, the good advantage is that it's possibly less prone to mutations because you're dealing with the whole virus with many antigens. Now, there are also disadvantages of this approach. First of all, this is long and expensive because you have to grow a virus in cell culture, and it requires live viral culture. So it requires very high standards of biosafety. So we call them biosafety level 3. You probably all know that this is very expensive. Not every country has these facilities. So you have really limited number of institutes that are able to produce it. And then also the disadvantage is that it requires growth systems, and meaning that in the vaccine, you can have the residual amounts of many things that you use to actually grow and inactivate the vaccine. And this is the contrary to the RNA vaccines, which we're going to be talking about later. So now for the traditional approach, for the inactivated virus approach, we have several companies and states which have already produce their candidates, and first three of them are from China, so Sinopharm, Sinovac, and Wuhan Institute. So Sinovac is already used in many countries. We have also Bharat Biotech, which is from India, which is going to produce many doses for Asian countries. We also have the vaccine by Chumakov Center in Russia, is by Russian Academy of Sciences. And probably will be interesting for you that French company Valneva is having one of the candidates of this type as well. Now, good news for us as well is that one of the candidates which was stated to be undergoing the third phase trial, as you know, is the vaccine COVID from the Research Institute of Biological Safety in Gvardievsky in the south of Kazakhstan. And actually, this is not, excuse me, this is not surprising for me, because this institute have been known for producing inactivated vaccines especially against veterinary diseases. And we have a big history of vaccine development in a way of biosafety concerns as well since the Soviet times. And I wish lots of luck in the third trial for this vaccine. Although I must say that I don't see a lot of data coming out. I would like if you or anybody who hears me now could transmit this information, that really the scientists would like more data about the first and second trial, about the results, in order to judge how it's going to perform in the third phase trials. Okay, so now let's move to the second type of vaccines. This is viral vector vaccines. And these are the vaccines, the Sputnik vaccine, the AstraZeneca vaccine, very famous candidates, which we're going to talk about. But how does it work? So basically, the idea is that we have our coronavirus, and we have the RNA, which is responsible for producing the spike protein. And we have adenovirus. Adenovirus is basically a common cold virus, right? We meet with adenoviruses every season. About a third of viral respiratory infections is caused by adenoviruses. These are the viruses which are very close to human. And basically, there were already vaccines which were developed based on the adenoviruses, for example, vaccines against Ebola, etc. However, this is the first time where we have the mass production and mass use of viral vector vaccines, which is actually good news for me as a virologist, because we have been speaking about them already since a long time. Now, basically, adenovirus was unarmed, so it became almost harmless. And what the scientists did, they have inserted this sequence of spike protein into the adenoviral genome. Adenoviral genome is a DNA, and basically what it can do, it can actually inject his DNA into the nucleus of a host cell, of a human cell, and then this is going to be produced. And among the genes that are there, so the mRNA, which encodes for the spike protein of the coronavirus, is going to be produced as well. What happens then? So now we have this RNA. This RNA is going to actually make inside the cell little spike proteins, which are going to be, so three spike proteins, they actually make up this spike, which are going to be exposed on the surface of the cell, and then will be followed by the immune response that we already have discussed. And when we speak about viral vector vaccines, there are basically a number of candidates already. I would like to speak about 4 of them. So first of all, let's speak about Sputnik, right? So the idea of the Sputnik vaccine is a combination of 2 adenoviruses, and these are 2 human adenoviruses, adenovirus 5 and 26. And the good thing about this is that they use 2 different viral vectors, and this actually reduces the ability to to reduce. So basically, the problem with adenovirus is that immune response that we produce against the vaccine is not only against the spike protein of the coronavirus, it's also against the vector itself. So when we use the same vector twice, of course, we would expect that immune system is going to basically destroy the vector straight away before even mounting the immune response against the coronavirus part. So that's why the Gamaleya vaccine, they have chosen to have two different adenoviruses so that they can reduce this risk. However, these are the viruses which could be met already in the population, meaning that the people who already have had the disease, they had a cold caused by this adenovirus 5 and 26, they will not respond to the vaccine. So this explains actually that what we expect as scientists from the viral vector vaccines is the efficiency around 60–70%, I would say. So that's why I don't really trust the numbers, the official numbers of 91%. So we can speak about it a little bit later. Let's speak about AstraZeneca, Oxford vaccines. So there's a major difference between the Russian vaccine and the Oxford vaccine is that as a vector, they use adenovirus, but not human one. It's the one of chimpanzees. And basically, also there were news recently that Oxford and Russian vaccines, they are launching the trials of how we can combine the two. And this is probably very good news because for AstraZeneca to be efficient, that'd be probably nice to have the vaccine by another vector for a second shot or vice versa. So this is good news for us as well because of the price, of the availability of the vaccine, and we would like to know, we would like to have some information of whether we can combine these vaccines. Let's speak about Johnson & Johnson and CanSino. The good thing about these vaccines that they don't need two shots. They need only one shot. However, Johnson & Johnson, they are starting the trials with two shots as well, just to double check that it's not giving any better results. And here you can see that, of course, for Chinese and the Russian vaccine, you had already early approval since June and since end of August, which is not really something that we usually do in vaccinology, right? This is really unprecedented situation. And I don't like it. And I was criticizing it since September because it's not because it will cause specific damage, health damage, because these are the vaccines that we already know, approximately we know what to expect. And by results that we see now, we don't really see so many side effects, right, of this vaccine. But the problem is our perception of vaccinology and our perception of how we test the safety of the vaccine. So this actually, we're going to work a lot in order to take back this respect to how the clinical trials are being made, I think, after this pandemic. Okay, now let's speak a little bit about this, some errors which were done during this trials and some of the communication errors, I think. So in AstraZeneca trial, you know, did anybody of you hear about this dosing error in AstraZeneca? You can answer. You can turn on your microphones. I'm gonna just double check that you're not sleeping. Кто-нибудь слышал про ошибку AstraZeneca, почему у них была ошибка во время клинического испытания? Кто-нибудь слышал про это?
Нет, не слышали.
Да, я тоже не слышала.
Не слышали? Vous avez entendu, il y avait une erreur chez AstraZeneca pour ce vaccin. Oh non, mon français ne marche pas aujourd'hui. Ok, смотрите, получается, okay, I'm going to speak about it a little bit in more detail. So basically the trials were done in the UK. First of all, they started in the UK and then they continued, and mainly the main outside cohort was in Brazil. And when you remember when everybody started to say that we have efficiency of 94, then we have efficiency of 95, we have efficiency of 91, etc., etc. So, you know, they started measuring their efficiency every 2 days. And then AstraZeneca came out and said that, okay, we have efficiency of 70. Then when scientists, so people started to look closely into these results, and they realized that it was 90% efficiency in Brazil trial or in some part of the Brazil trial, and like 60% or something like this in the UK. And of course, that was a good question why it happened. And they realized that they actually made a mistake in Brazil, having the first half dose. So there were 2 injections, right? And they, by mistake, I don't think it was a mistake, they put half a dose in some of the population cohort in Brazil. And they saw actually that this half a dose on the first injection was actually working better than a full dose. That can be explained very easily by the nature of this vaccine, because as I said, we don't want immune system to be overactivated against the vector. And when we probably when we have a half dose, our immune system is going to be just primed just enough in order to be boosted on the second injection. So, and this was not really responded well by the AstraZeneca officials. They didn't really open all the data. They were just like, you know, we're gonna publish. Yeah, we did a mistake. It was a dosage error, and we're gonna publish it in the scientific paper, and that's it. So they didn't really comment on this, and I think that was a major communication mistake, because I think this is something that will that will cost them more in terms of registration. Всем понятно на русском объяснить? Получается, там была такая типа ошибка в кавычках, да, вот, и в итоге AstraZeneca, они не поделились данными, они не открыли свои данные, в принципе, до сих пор не очень понятно, почему это произошло, и поэтому сохранилось к ним недоверие. Хотя это были хорошие испытания, да, но они, как бы, когда такая ошибка обнаруживается вообще, в принципе, да, испытания должны приостановиться, они должны начаться заново, если они говорят, что это та формуляция, которая работает. Но, конечно, этого не произошло, потому что как бы мы во время пандемии. Да? Поэтому, когда мы говорим об ошибках, это не только российские вакцины там, или китайские вакцины, или наши казахстанские. Практически все производители, у них были такие, особенно ошибки в коммуникации. Да? Okay, now let's speak about Sputnik vaccine on the other hand, right? Another vector vaccine. So the problem with this vaccine and with this third phase trial is that this trial will never be finished. Why? Because the people, so as you know, for the third phase trial, we have the group of vaccinated and the group that received placebo, right? And usually by the standards, by the high standards of the clinical trials, this should be double-blind, placebo-controlled trial. What does it mean double-blind? Nobody knows whether they received placebo or vaccine. And the problem was that people started to check their antibody titer, and they started to post it online on a Telegram channel. And because the people who were vaccinated, and they actually did have good antibody titers, and they had a certain range, and they could actually tell that if in 2 weeks they have certain range, they were vaccinated. People who had placebo, they realized, they also did the tests, and they realized that they're having placebo, and they went to make a vaccine. And that's why we have a big mess with the results. And these results can never be published because basically this trial is not finished. They should be monitored at least 6 months after the completion of the second dose. And because they already started to measure the antibodies by themselves and they went to vaccinate themselves, they cannot be treated as placebo, so we cannot treat the results of this trial valid at all. This is, of course, bad news in terms of science, but it's actually good news in terms of understanding the real efficiency of this vaccine, because as we know, officially it's 91% efficiency, but there is this Telegram chat where you have all of the people, like already, I think more than 6,000 people who have reported that they were vaccinated or they had placebo, and then they are posting all their results about antibodies. And by this Telegram chat, we can say that the efficiency was about 70%. So this is good news in a way that internet actually, it helps us to understand the real efficacy of this vaccine. However, of course, it's not funny when we speak about science, right? Okay, now let's speak about the newsmaker in this race, excuse me, the RNA vaccines. So these are my favorite vaccines. Why? Because when I went to Institut Pasteur for a vaccinology course, and it was in 2011, and we were speaking about RNA vaccines like something in the future, like really something that would happen probably in 20 years. And I'm so glad that we're actually seeing the first RNA vaccines produced during this pandemic. So what is the RNA vaccine? It's basically, so we know that this is a viral RNA. We have the part of this spike protein gene, and basically some parts of this spike protein, spike gene, we pack into lipid nanoparticle. It's like a soap bubble, you know. It's basically RNA in a bubble, and nothing else. And that's why it's so brilliant, because it's simple and elegant. It's absolutely clean production. It's based solely on chemical synthesis, so it doesn't require cells, it doesn't require yeast or any of the things that bother so many parents in vaccines, even though they're relatively safe. But still, with this cleanliness of production, we actually can achieve exceptional safety, I think, with the years coming. It's also quite easy to adapt to virus changes, because we can just change the sequence of this RNA if the virus has mutated so much that the vaccine doesn't work against it. And when we speak about this, the Pfizer, no, it's not Pfizer, BioNTech CEO, he was saying that if the virus changes so much that the vaccine cannot protect, they are able to produce 1 million doses in 2 weeks of the new formulation of this vaccine, which is pretty cool, right? And then the last thing, that which is not least, is large scale possible with these vaccines. And also this is about the number of doses. They're speaking about 1 billion doses already this year. So this is something that we could not imagine before. And this is the technologies that makes it possible. Now, there are, of course, some major drawbacks. And first of all is transport and storage. Probably you've already heard that, you know, RNA is a molecule which is very fragile, and it's supposed to be stored in the lab. We store it at -70. If you think about -70 freezers, this is quite a big machine, which requires a separate room, which requires separate coolers. It's a bit expensive. It's not present in some, like in all countries, and this is really a challenge for the manufacturers. But Pfizer, for example, they have made some special boxes with dry ice and special, you know, procedures in order to make it possible. Moderna vaccine, on the other hand, they have changed their technology in a way that it actually can be stored at higher temperatures, which is also quite nice. We're going to discuss it later. Now, the major drawback for me actually is that it's completely new technology. And with these new technologies like this, so we have never used RNA vaccines on humans. And we have to be very careful with side effects. So this is what happened with this allergy story, you know, that allergy people with a history of severe allergies, they were not included in Pfizer trial. They were included in the first day of vaccination in England, and bam, we had like, I don't know how many, 2 or 3, well, anaphylactic reactions, right? So this is something that we really need to be careful about, even though that we expect that this is a vaccine that is probably safer than all other types of vaccines. This is something new. And I think that this 2-year post-registration monitoring is a very good idea. Okay, let's, what happens with this RNA? How does it work? So basically, this RNA, it enters the cell, and there it starts to use the cellular machinery in order to produce the spike proteins. And these spike proteins, they make the spikes, which are being exposed to the cell surface, or it produces the part basically which will mount the immunity. Now, when we speak about immunity, we know that in adaptive immunity, we have 2 major branches. It's antibodies, which everybody knows. So the antibodies which actually bind the virus, which don't let virus to proliferate and to enter the cells. And we also have T-cell immunity. In this case, we have killer T cells, which which doesn't bind the virus but kills infected cells, which exposes the antigen, which is also quite important. And it was shown that in the case of coronavirus, when we speak about memory, the T cell immunity is probably even more important than the antibodies, but because the antibodies titer doesn't really correlate with the memory, so with how long the immunity lasts. Okay, so now, oops. Okay. Yeah, this are already. If you have questions about RNA vaccines, don't hesitate. So these are the two major mRNA vaccines. So the Pfizer BioNTech and Moderna, they had quite a good number for the trials, number of people, I mean, and the main difference here is the storage, of course. Moderna had finished the trials a bit later, and they're still they're starting this post-registration story. However, they have a very good advantage of temperature, of course. But I personally, this is also my own opinion, I really like how Pfizer vaccine was made, because the idea is that they started to think about RNA vaccine against coronavirus already a year ago. So they really saw that this is going to be a pandemic very early, and they have started to work at it straight away. And already in May, they started the third phase trial, can you imagine? So, and they really had a good, nice trial and very, very transparent reporting of the results, which I really, really like. And this is thanks to these two people, because this is also a new example of how pharma development is going to be changing, actually, because we're not speaking here really about big pharma. Of course, there is a Pfizer, which is responsible, of course, for the success of the third trial, for the investment, and for the achievements in the transport chain. However, this was started as a startup, and it's an example of immigrant science, which is quite important, which has made headlines. And for me personally, it's also quite important as an acknowledgement. And it was really done by scientists. So these are the scientists who really have good publications and who are not on the side of CEOs and commercial side of pharma, but really on the side of science. And this you can really see when you hear to them answering the questions of journalists, etc. Right? I really like this new paradigm in vaccine development. Now, okay, now let's move to the last type of vaccines, which I'm going to speak to you about, is protein-based vaccines. And here I would like to speak about one example, which is Novavax. This is also this example of a small company, and nobody heard about Novavax before, and they haven't produced any product before, but they are one of the leaders of this race. So what is this vaccine? Basically, we're speaking about baculovirus, and baculovirus is a very nice, elegant biological system. It's one of big viruses which are able to enter the cell, and inside the cells they can produce the part of the DNA that is encoded in them. And funnily, this is produced in moth cells, and these are moths. So basically, some butterfly-like creatures, which cells were using to produce the proteins of the spike. So the spike proteins of coronavirus, and then basically the spikes which we produce in moth cell in this Novavax vaccine, they are building this nanoparticle. So several spikes are building one nanoparticle, and they have been added to immunity priming compound with some plant-derived compound, and this acts as adjuvant. So basically, it acts to awaken the immune system. So together, this is a protein vaccine, which is Novavax, and as well, it can give rise to T cell immunity and to B cell antibody mediated immunity. So this is also quite nice because it's new, and it's a nice expression system. It's very nicely thought through as in terms of science, right? So Novavax, it's a big surprise actually, because this is a small biotech startup which was not known before. They have started the trial in October, but they have very promising results, and they have secured more than 1 billion doses. Can you imagine? Another protein-based candidate, it's a Russian candidate, Epivac Corona, which was already approved for limited use in Russia as well as Sputnik, and they are, I think they started already mass vaccination in January with this one. So what I do not like about COVID-19 vaccine development, I don't like this efficacy claims. I don't I don't like this race in the style of Cold War, because as you remember, Pfizer, they first started to report their results already 1 week after the second dose. This is not how it's usually done. Usually we need minimum 2 months, but better 6 months before we can actually report something about the efficacy of the vaccine. And then it was followed by Moderna, it was followed by Gamaleya, by AstraZeneca. And I would like to emphasize that we really need scientists to speak about this efficacy data, not CEOs and not presidents. And I think it all goes a little bit in a question of the commercial use, of course, and this is normal because this is pharma, but as well, when we speak about the new pathogen like this, I would like that we focus our attention on the science. And what I do like about vaccine development during this pandemic is that there was a lot of science and there was a lot of work. And when we speak about all of these candidates, the scientists that have worked behind these vaccines are really good scientists, and that was a good quality data for most of them. And of course, I was very glad to see first ever RNA vaccines, and and also lots of investment into new vaccine technologies. I can shortly speak to you about some very funny and surprising vaccine candidates. For example, the company Philip Morris, they're known for the cigarettes, and basically they're also producing a vaccine. I don't remember the name of the company so they're working with them. And basically the virus is grown in tobacco cells in order to produce it, and it's quite nice. There are also one candidate where a vaccine is done in a form of a skin patch, and they have 600 nanoneedles which transmit the DNA of the coronavirus, and it's really, really cool in terms of technology. There is also one very nice trial which studies the role of BCG vaccine against the coronavirus, because we know that BCG was found to have so it's the effect of trained immunity. So in Australia, there are 10,000 medical workers which are receiving this vaccine in order to, so BCG vaccines that we all know about, to actually see whether this helps against the coronavirus disease. Okay, now I think, let's see what else I have. Okay, right. Now let's speak about how fair it is in terms of mass vaccination. Of course, we have this concern that not all of the people have the same so they don't have access to vaccines. Not all of the countries have the same access. And of course, the example of mass vaccination is probably Israel, because they already have given at least 1 dose of Pfizer RNA vaccine almost to 1/3 of their population. By the number of doses, the leader is the United States, which is also not surprising. However, of course, I think it's our goal eventually is that the access will be more or less even, but it's not achievable at least this year, I think. Now let's probably discuss more about the questions that you have. I didn't give my opinion of what is the vaccine that I prefer in terms of whether I can advise any. I don't know, but because I wanted to hear your questions first. What questions do you have about vaccines? was everything clear? And maybe I need to explain something in Russian or something.
Estelle, thank you for your presentation. Do you hear me?
Yes. Yes.
So my question is about the safety. So what do you think about the efficiency and the safety of the results of the Sputnik V? Because I mean, they had so many issues related to GCP and related to ICH requirements.
Okay, for the safety of gamma layer vaccine, basically what they have in their official data is that there is a number of side effects which are normal for adenoviral vaccines. Because when we think about adenoviral vaccine, this is basically a cold virus, and we expect to have more side effects, for example, in the elderly, as opposed to healthy young population, because this is it will act as a cold virus, right? And that's why about 90% they have temperature rising, they have fatigue, 80% I think it was reported to have headache. So this kind of reactions, they are normal, especially for adenovirus vaccine, especially for adenovirus vaccine which was made this way and which was made this fast. So I think for me personally, I didn't see any surprises in a way, like for the side effects. But of course, the concern is, what data do we have? What data do we see? The official data for the efficiency, of course, it's more than the actual data. I think 91% is too much for adenovirus vaccine. It's not even realistic when we think about it. So what we expect is 60, 70%, because many people have already met with them. So when we think about this, if we cannot trust the efficacy data, we probably cannot trust the safety data. However, all of this spill of information into Telegram chat, it gives us actually the actual information about the safety and efficacy. So as I said, from there, we can see that most of the people have temperature, headache, fatigue, etc. for maximum 2 days, and that's it. So from the actual record of people who were injected, and there were a few thousand of them, we can see that there were no surprises in terms of safety. We can see that the efficacy is less than the official data, but for me, I mean, it's fine. It's something that I expected. Yeah. For the standards, for the standards of safety, for the laboratory standards, I cannot really tell you much, and I also cannot tell you much about the manufacturing plant which is going to be built in Karaganda. I don't have information about this, so I better not.
So this is actually generating other questions from my side, and my question would be like this. I mean, we know that the adenoviruses is actually, well, quite usual for our, populations. They're already circulating in it. So they are using I know that there's some subtypes of the adenoviruses that are actually quite rare, but despite the fact that they are rare, they're still actually circulating in society. So that means that majority of, some small group of population would be already be already immunized. Yes?
That's it. So they're going to be immune to the vaccine. It means that the vaccine will not work for them. That's why we expect lower efficiency for the vector vaccines, which is normal. Because even if it's rare strains of the adenovirus, they still share some similarities with other adenoviruses. So do you see? So that's why we really, it was not very clever to speak about more than 90% of efficiency, because biologically we expect that the efficiency will not be more than 70%. So that, yeah, I don't know if it answers your question.
Thank you for your reply, Asel. Thank you for your reply. I mean, I would like to say that this sort of issues actually were seen in AstraZeneca vaccine. So that is why I just simply assumed that they would have this very similar efficacy.
They would just, the good thing with AstraZeneca is that they have made claims which are a bit more realistic.
Yeah. I mean, they used a very different vector as well, because they
but this
used
is chimpanzee vector.
Chimpanzee, yeah.
Yeah. Yeah, they use chimpanzee vector, which is good in a way that it's a bit like it's more different from the human. Yes, but still, as I say, that when we speak about the viruses, which normally the group of viruses which circulates in the human population, we would expect some immune response, immune reaction on the vaccine already. Yeah, even if it's chimpanzee virus.
Also, if I may, about Sputnik 5, you discussed some of the problems with the testing and so on. And my question is, do you see a clear path for registration and certification of the vaccine in EU and UK? And I mean, as Hungary, I guess, have certified it. And the reason for the question is very simple. At some point, we're going to arrive at passporting of vaccines, and travel to EU and UK will depend, I guess, on whether a particular vaccine is registered or not.
I think it was claimed by EU officials. I don't know. I have seen this information about a week ago that the passport of the vaccine will not be the obligation for having Schengen visa. So I hope.
No, also, I think it's a little bit, if I a little bit interrupt you here, it's not. It will never be a requirement, but there will be differences where you are treated. If you don't have passport that's accepted, you're gonna have full quarantine, self-isolation, and so on. If you do, I assume this is speculation, but I think this is where it's going.
Yeah. But I mean, about Sputnik vaccine, I don't think it's going to be accepted widely in the Western countries. But the main market for this vaccine is, as you know, developing world, Arab countries, which have or South America, which already have secured many doses. So I think market-wise for Sputnik, there are no problems. For the people who have vaccinated, probably. Although I think if AstraZeneca and Sputnik, they have started recruiting people for the combined trial. And I think if there's going to be people who are injected by the two of them as a combination, that would probably be a decision for some of the people. And I think there are also going to be trial whether we can combine vector vaccines and RNA vaccines. And this is another possibility because some people, they want to be at least protected with one dose. And that's why they go on to vaccinate with Sputnik, because this is the only possibility. And then they say straight away that, okay, and then in 2 months, I'm going to take Pfizer, because it's going to be available. So this is one of the possibilities.
But what you're saying is that it's really hypothetical, and that before it's tried, it's not a good idea to vaccinate with two separate vaccines before it's understood how they interact.
Of course, because we haven't used vector vaccines before. We haven't used RNA vaccines before.
Okay.
I don't think there is a good idea. Also, when we speak about vaccines, when we speak about side effects, we usually speak about first 2-3 days or a week. We usually don't, like, during the 3 phases of clinical trials, normally all of the long-term kind of effects. So there is not going to be normally any serious long-term effects. However, as we see with Pfizer, as we see with Sputnik as well, there are certain cases where we didn't expect, for example, RNA vaccine to give this anaphylactic reaction. We really didn't expect that because there's not much in there that could cause this, but still we got it. So I am in no right to say that it's okay to vaccinate with 2 of them before we have any data.
Thank you very much.
Okay, I see 2 questions in Russian here. Когда и где возможна вакцинация в Алмате? Какая вакцина будет доступна? Я думаю, что будет доступна сначала Спутник 5, и она уже будет доступна, насколько я знаю, с 1 февраля. Но там, получается, есть какая-то определенная очередность, то есть сначала медицинские работники, учителя, да, и потом дальше. То есть там нужно уже смотреть. Здесь я информацией особо не владею. Я думаю, что мы будем, в принципе, на этой неделе мы узнаем больше. В течение какого времени будет стойкий иммунитет после вакцинации? Это очень хороший вопрос, потому что мы, в принципе, не знаем, даже инфекция от коронавируса, она дает ли вообще иммунную память. То есть сейчас у нас есть данные, что те люди, которые переболели в легкой форме, у них антитела длятся, то есть они сохраняются около 3 месяцев, к сожалению, и потом они, есть как бы много уже таких случаев, да, которые показывают, что после 3 месяцев не у всех они сохраняются. То есть, в принципе, переболеть повторный раз – это реально, очень реально, особенно для людей, которые переболели в легкой форме или бессимптомно. Люди, которые переболели в тяжелой форме, было показано, что у них антитела на таком нормальном уровне сохраняются около полугода-год. Вообще, когда мы говорим об иммунной памяти коронавирусов, да, это нормально для них, что как бы иммунная память недолгосрочная, потому что эта группа вирусов, она тоже нам знакома, она тоже приходит к нам каждый простудный сезон, и было показано, что даже уже через 2 месяца можно повторно заразиться тем же самым коронавирусом. Это определенная особенность коронавирусов. Здесь, кстати, к вопросу о том, что нужно ли тем, кто переболел, повторно вообще вакцинироваться, да, имеет ли смысл? Смысл имеет, и для этого вам нужно, если вы думаете, что вы переболели, вам нужно измерить у себя титр антител, и посчитать примерно, когда вы переболели, сколько времени прошло. Если, например, уже прошло там 4 месяца, полгода, то, в принципе, не лишним будет вакцинироваться. Потому что повторное заражение, оно не обязательно легче, чем первое. Были уже случаи, когда оно было тяжелее, да. Если вы переболели один раз бессимптомно, это не значит, что вы второй раз будете болеть тоже бессимптомно, потому что здесь это зависит от инфицирующей дозы. То есть, например, первый раз вы могли переболеть бессимптомно, потому что вы ходили в маске, или там потому что, была маленькая доза вируса, да. Вот, второй раз, если вы встретитесь, не факт, что ваш иммунитет вот этот довольно маленький, да, потому что количество антител маленькое, или там Т-клеточный иммунитет, вот, что вы можете переболеть так же легко. Вот, поэтому это к вопросу о том, стоит ли вакцинироваться переболевшим. Публикации по данной теме, да, вот вопрос, антитела сохраняются на нормальном уровне. Тут, получается, есть определенный титр антител, я могу вам, да, прислать публикации, то есть сейчас есть уже, таких вот маленьких публикаций, препринтов очень много уже по измерению антител. Я считаю, нормальный, там есть где-то 2-3 статьи, которые говорят о том, что там как они измеряют антитела и сколько времени это все продолжается, да, вот, поэтому там зависит от уровня, от титра, то, что вам покажет анализ, и вы можете уже судить, насколько у вас они еще сохранились. Потому что этот титр, он меняется в динамике, да, то есть если мы говорим о том, что, скорее всего, против коронавируса не будет пожизненного иммунитета, если вы переболели. Вот, публикация имеется, я могу прислать потом. This is just to repeat in English that I was speaking about the question of immune memory in coronaviruses. And we know that coronavirus is a group of viruses where immune memory is quite short. And this is not surprising for us. We already know it since several years, that coronaviruses that are common cold viruses also, so the person can be reinfected already 2 months after having the infection. And regarding this, of course, there is some information that people who have had the coronavirus as in an asymptomatic form, or who didn't have lots of consequences, however, they would have lower titer of antibodies and shorter immune memory, most likely. So the people who have had the need of oxygen, who were hospitalized, they tend to have more antibodies, and they tend to have basically longer immunity. However, it also lasts, it may last a short time, about 6 months. So this is to the question whether the people who have already had coronavirus need to be vaccinated. And the answer is, it depends really on your state of immune memory. You can measure your titer of antibodies. You can probably make some judgments about this. You can also just probably calculate when you think you had it. If you had it like in March, it's probably a good idea to be vaccinated now. Окей, вы бы себе и своим родным вакцину какого производителя сделали? Смотрите, это такой вопрос очень сложный, да, я поэтому на него специально не отвечала в презентации, потому что это решение для каждого человека, да. Здесь, конечно, из всех кандидатов, которые есть сейчас в мире, наверное, Pfizer это самая проверенная вакцина, да. Это проверенная вакцина, в том числе и потому, что там, скажем, в Израиле, да, мы видим уже огромное количество вакцинированных в европейских странах. То, что мы видим из побочных эффектов, это касается или очень пожилых людей, которые в принципе, да, реагируют. Вот те случаи в Норвегии, да, я надеюсь, что вы понимаете, что в Норвегии 40 человек ежедневно умирает в домах престарелых, да, и поэтому 13 смертей после вакцинации – это просто 13 смертей в доме престарелых, да. То есть, по сути, та информация, которую мы сейчас получаем о Pfizer, она открытая, во-первых, да, во-первых, она реально очень большая открытость этого производителя. И мы видим, что таких каких-то больших сюрпризов с точки зрения побочных эффектов мы как бы не видим, да, не наблюдаем, это очень хорошо. Поэтому если бы у моих родных был выбор, то, конечно, я бы советовала Pfizer. Но дело в том, что у моих родных нет выбора этого, как и у вас всех. Как и у меня сейчас, да, потому что я сейчас совсем не первая в очередь, даже несмотря на то, что я во Франции живу. И поэтому, когда мы говорим о людях, у которых большой риск при заболевании коронавирусом, то есть это диабетики, это люди с ожирением, люди с сердечно-сосудистыми заболеваниями, люди с хоу, с заболеваниями почек и так далее, да, то есть мы можем ожидать, что этот человек в силу возраста или в силу риск-факторов может реально серьезно заболеть коронавирусом. Мы знаем, что сейчас идет следующая волна, и мы знаем, что наши больницы, они не справятся, как и не справляются все остальные больницы во всем другом мире, да. Поэтому в данном случае, наверное, будет правильно вакцинироваться той вакциной, которая есть. Потому что то, что мы видим, опять же, по спутнику, да, это я не говорю сейчас об официальных данных, да, потому что официальные данные, они не слишком открыты. Я говорю о том же самом Телеграм-чате, вы можете туда зайти посмотреть. Вот все эти побочные эффекты, то есть гриппозные симптомы, да, в течение там 2 дней, это норма для аденовирусной вакцины. То есть мы опять же не видим там каких-то больших сюрпризов. Поэтому с точки зрения безопасности, в принципе, спутник 5 на данный момент, по данным, которые сейчас есть, она приемлемая вакцина, и особенно в условии, когда нет другого выбора, да. Вот, поэтому вот такой ответ. Точно так же мои все родные живут в Казахстане, точно так же я им отвечаю. Вот. This is a very difficult choice. This is really not an easy question. Да?
I still have another question, and this is actually the last question. I know that I'm extremely annoying, so yeah, I would like to actually be not annoying so well. So my question is related to PCR diagnostic of the SARS-CoV-2. So I just recently received a lot of different sort of emails and calls from my colleagues that are working in medical fields, and they, stating that they have faced it with a lot of cases of pneumonia, and this pneumonia actually have a lot of signs of, many signs of SARS-CoV-2. But the thing is here that the PCR is negative. But and my question is actually, what do you know, or maybe you are aware about that? That what sort of antigens actually using our laboratories in order to actually simply, confirm the SARS-CoV-2 in Kazakhstan? Because I know that in UK and US they are using 3 antigens.
Yeah. Actually, it's quite an interesting story in the UK, because you know this British mutation that have recently been found since mid-December. They have found about it because of the PCR tests. They have used 3, so the DNA, the RNA sequence from 3 proteins, encoding 3 proteins, and the sequence in the spike protein, it was in this binding domain, which actually binds to this door. You remember I showed you the ACE protein. And basically what they saw in the positive samples, that 2 of the bands were there. So you should have 3 bands on the gel, right, when you do PCR, when you do this test. And the 3rd band was always absent because it was not there, it was not detected by the test. But the other 2, they were. That's why they knew that this is coronavirus, this is positive PCR, but this band is absent. And this is how they discovered that it was actually this mutation in the spike protein, which now is prevalent in the UK, and it rapidly, I think it will spread everywhere, and it gives more, it's more contagious. So basically, yeah, that's why actually I know that yes, the UK tests, they do these 3 different regions of the RNA. I don't know what tests are being used in Kazakhstan. I think also the problem is how we count the infected, because one thing is PCR. Of course, PCR is important, and PCR is used in most of the countries in order to count the number of of the infected people. However, now I think we are in the position where we can give clinical diagnosis of COVID. I think in March we couldn't give the clinical diagnosis without the PCR, because we didn't know as much about the symptoms. And now I think we're in the position where we can actually, even not having the PCR positive results, because you know, the ratio of negative results is about 40%, I think, not even 50 in some countries. So it means that half of them are false negatives. And that's why I think we need to think about clinical diagnosis in this case, because we already have enough information to give this kind of diagnosis. But this is very complicated in terms of how it's going to be controlled by the government, and how we're going to judge our statistics and stuff like this, you see. Because then it also the count, not only the count of infected people, but also the count of deaths. And we will have much more numbers, you know, much bigger numbers for the deaths if we count this clinical diagnosis with false negative results. Because PCR, first of all, it depends on the machine, it depends on the test that you use, and depends on the proteins that you study. Yes, it also depends on when you do the test, because there are many studies which show that, I mean, if you do the test like 1 week, or I don't know, like if you do it too late, the virus is going to go down up to the lung, in the lungs, and you're not going to detect it in the nasal swab. If you do it too early, also you might not have enough of the virus, etc. You are speaking here about the infectious dose. So basically, the PCR test, that it's something that can be affected by many factors. I would think that actually the antibodies, basically the antibody test, the IgG test, it will give you probably a much clearer view. And probably this is something that countries should do in order also to decide on the priority for the vaccination, to study the antibody titer of the population. And this is a bit more clear than the PCR, in my view. If evolution of virus might make vaccines inefficient and therefore useless. Okay. Yes, so basically this is a question about mutations. It's very interesting. I wrote in Russian, I wrote a big article recently in Vlast KZ. You can all look. It's called Your Guide to Mutation, ваш гид по мутациям. Возможно, вы уже читали. Прочитайте, если вы не читали. Я думаю, что она очень интересная. Я потратила все мои каникулы на эту статью. So I spent all my New Year holidays writing this article. You really should read it. Basically, the mutations are normal for the virus. And it's normal that the evolution, so there is evolutionary pressure for the virus to change. And this is how viruses survive. So you can imagine that this virus, it jumped from an animal host. We can say if it's a bat or a pangolin, it doesn't matter. It jumped to a human being. And in the human being, inside the body, this is completely different conditions. And as you know from Darwin, that when we have different conditions, we're going to adapt. So first main mutations that coronavirus had, it was in the spike protein. It's called D614G. So basically, when you see all these numbers and the letters, you see the change of amino acid. One amino acid called D on the position 614 was changed to amino acids called G. And this change, actually, it makes this protein completely, well, not completely, but different in the shape of the protein, because amino acids are different by the chemical properties. They're hydrophobic, hydrophilic. So basically, the idea is that you have this, you have the ACE protein on the surface of the cell, you have the spike protein of the virus, and spike protein, it basically it's like a handshake, or we hold our door and we open it, and it opens the door inside the cell. So what happens when we change something? Basically, the spike protein, it's going to be changing so that it really it's more tightly connected to the cell, so that we can make the entry of the virus more efficient. That's why we say that that basically most of the mutations that coronavirus have had during this year, they have made it more contagious. But it doesn't mean that they made this virus more dangerous. And I will explain you why. Because in evolutionary points of view, this virus is not really deadly. Well, we see 2% of mortality rate as by numbers now. However, it's still not as deadly as many other viruses. That's why this virus, it doesn't have the pressure to become less deadly or more deadly. It's not basically the terms where so what we're speaking about when we speak about the mutations of coronavirus. We really speak about just adaptations. And this British mutation, however, this British mutation occurred in the region which is used by Pfizer vaccine. And of course, it was asked on the press conference, and the head of BioNTech, he said that in 2 weeks, we're going to have, if we need to adapt this vaccine, we're going to have it in 2 weeks. We're going to have million doses of new formulation in 2 weeks if we need to change this letter in the RNA. This is, of course, good. However, I think it's not that simple. It's just, so there was one paper, one preprint, which told that the Pfizer vaccine still works against one of the mutations, but it doesn't. We don't know whether it works against other, because this is the British variant. So that you know, it contains more than 23 different mutations, all in more or less the same protein, in the spike protein. It means that all of these 23 mutations together, they really change the shape of the spike protein. They really help, I don't know why, but they really help spike protein to become more efficient in terms of binding to the cell. Now they tested one of these 23 mutations and they say Pfizer still works. This is not the final answer, because we need to see whether Pfizer still works against this combination of mutations. And this is much more difficult to do in the lab. And actually, 2 days ago, there was another paper, another preprint published, which tells that Pfizer doesn't work against 3 of the mutations in this variant. Do you see? So I, as a virologist, I expect that Pfizer wouldn't work. I don't know. I think it shouldn't work as well against the British variant, because the British variant really changed a lot in the spike protein. So we're going to be waiting for the results, and maybe it's good news for us, because, well, in a way that vector vaccines and inactivated vaccines, for example, they're less prone to these mutations, because inactivated vaccine is the virus, the whole virus itself. And that's why we have more of antibodies which are produced against it, and more chance that the vaccine will still work against the mutation. But this is very interesting topic. I can speak about it a long time, and I don't think it's very interesting for you, probably. Do you have any other questions? Есть еще вопросы? Vous avez des questions? Okay, immunity we spoke about, antibodies, right? What else? You can speak up, or probably we can finish the presentation. I don't know.
Уважаемые участники вебинара, если у вас еще дополнительные вопросы, вы можете включить микрофон и задать их, так скажем, вживую, а не в чате. Если вопросов нет, то мы, пожалуй,
Добрый день.
Да.
Okay, I see one question here. Ой,
Меня слышите?
говорите, пожалуйста.
Алло.
Да, мы вас слышим.
Смотрите,
Очень плохо слышно. Наверное, может быть, лучше написать в чат.
что
Очень плохо слышно. Напишите, пожалуйста, в чат. Okay, I see a question here by Philippe. Are
Слышно
vaccines
меня?
useful?
Здравствуйте.
Было до этого очень плохо слышно?
Меня
Очень плохо слышно. Напишите, пожалуйста, в чат. Okay, meanwhile, I'm gonna answer this question. So for children and teenagers, they have not been used on this age, and we're waiting for the results. I think there are expanded trials that are going on. We have to really pay attention here, because on the breastfeeding mothers, on the pregnant women, on the children and teenagers, they have not been used. Some of the vaccines, I think they have some limited approval for breastfeeding mothers. However, the rest, the pregnant, the children, no. And I don't think it's a good idea to try. I don't think it's even allowed. It's not even legal, I think, for any of these vaccines, not yet. So we just need more data. Напишите, пожалуйста, вопрос в чат, и мы вас очень плохо слышали. Как звали девушку? I think it's not allowed for anybody below 18. Well, as far as I know, for Sputnik, for sure, I think it's not allowed because it has not been used. For the rest, I'm not sure. I have to check, but I think it's logical that it would not be allowed for the below 18. Кто-то хотел еще что-то сказать, говорите, не стесняйтесь. Хорошо,
Так,
тогда, наверное, мы можем заканчивать, да?
Да, я думаю, да.
Хорошо.
Асель, мы, наверное, вас отпустим, потому что мы знаем, что у вас очень плотный график. И спасибо, что вы все-таки уделили нам время. Еще раз благодарю вас от всех членов нашей палаты. И вообще и в прошлом году все очень так продуктивно прошло, и вы с большой готовностью всегда у нас отзываетесь на наши просьбы, вопросы. Вот, спасибо большое. Желаем вам плодотворного года. И
Спасибо.
надеемся, да, что все будет потихоньку налаживаться, ну и каждый сам примет для себя решение использовать вакцины, либо нет, если использовать, то какие. В общем, надеемся, что все будут здоровы и счастливы.
Спасибо, Асим. Да, главное, чтобы это решение было информированным. Поэтому, когда мы информированы, я думаю, мы можем...
Да, самое главное, да. Информирован, вооружен.
Спасибо
значит.
большое. Да. Спасибо большое, да. Всем хорошего года.
Всем всего доброго, да, и плодотворной, хороших выходных. Всем до свидания.
До свидания.